Okay, so we'll start with the announcements. We've got two announcements today. There's tomorrow at 11 o'clock in room 102. There's a CAO seminar by Gutum Charteri from Bar Ilan. We'll be talking about challenges in observing aerosol cloud interactions in satellite measurements. On Sunday at 10:30, Yuval Moner will be talking about revealing hornblende brittle versus plastic behavior with implications for the rheology and isotropy of the lower crust. And now we're going to introduce our speaker today, Nimrod Schwartz from the Department of Soil and Water Sciences at Faculty of Agriculture in Rehovot. He's also the head of the department. He did his bachelor's in Rehovot and then did his master's and PhD at the Technion. He then went on to do a postdoc in Belgium before coming back to the Department of Soil and Water Sciences. And he'll be talking about root studies.
Yeah. Okay. Thank you, Emmanuel, and thank you all. So I'm going to talk today about the hidden half, about the root system, and how we can use geophysical method, and mostly I'm going to talk about geoelectrical method, to monitor root, to measure root in the subsurface. Okay, so a short and maybe unnecessary introduction and motivation. So I think it's quite clear to everybody that root systems are important, right? So they provide physical stability to plants. They enable water and nutrient uptake. They sustain crop production. They are part of the water cycle, of course, carbon cycle, and so on and so forth. So really, it is hard to overestimate the importance of root in the ecosystem. However, it is really hard and challenging to measure root in the subsurface. And the main reason is that we cannot see them, right? So the soil is opaque. It is really hard to have direct observation of the root system. So this is one problem. Another problem is that root are and soil. Those soil and root are extremely heterogeneous bodies. So the spatial heterogeneity is huge for soil, as you probably know. And of course, water content, salinity, nutrient, they are all dynamic properties that change in time. Root also, they grow in the soil, so it's really hard so really heterogeneous system. And the current method that we have, so the most traditional established method is coring, so just to take soil or root samples. And of course, this is destructive. We cannot go back to the same location and sample again. It is also expensive if you want to sample in many locations. And of course, it's really hard to take into account this huge heterogeneity and dynamic that I'm talking about when we make some local samples. We can also install sensors in the soil and to monitor, for example, water content or other properties. We can install camera and to look at root, but those methods are localized, so we can get information, local information, but we cannot see the huge heterogeneity. There are other methods like CT, MRI, in which we can see the whole system, the whole root system, or neutron radiograph. But typically those are small scales, they're expensive, and it's really hard to upscale the information that we obtain from those scales into the real life scale, so to the field scale. And this is where we can use geophysics. So, as I said, the traditional method are destructive, labor-intensive, slow, expensive, and so on. And geophysical method, okay, they are non-invasive in nature, right? And they designed to give us spatial map of the subsurface distribution. They are relatively low cost, and And this is like the main reason to use geophysical method to explore the subsurface. So I'm working with geoelectric, and the main geoelectrical method to map the heating half, to map the root system, are electrical resistivity tomography, ERT. And in this method we measure the resistivity, which is sensitive to water content, salinity, the pore geometry. The second method is induced polarization. In this method, we measure the frequency-dependent conductivity, or the admittance. Many people use impedance, so this is just 1 over the impedance. And typically we do it at a relatively broad frequency range. In application in soil science and geology, typically the maximum frequency is around 1 kilohertz, something like that. And the last method is self-potential, in which we can monitor the potential, the natural potential field developed due to electrochemical potential gradient or flow in clay, so streaming potential, but I'm not going to talk about this method. We are not using it to look at it. So a few words about ERT. The idea is simple. We apply electric current. I cannot see it, I'm colorblind. So we apply current between 2 current electrodes, and we measure the potential difference in 2 potential electrodes. So really a very simple method. We do it many times, okay? We want to get a map, not just one measurement. So we repeat this current injection and potential measurement at different locations, and we can do it in different arrays. So you can see here example to the Wenner array, where the current electrodes are here and the potential electrodes here, or dipole-dipole array, where the current electrode here and the potential electrodes here. And in a classical or standard ERT scheme, we repeat those measurements many times. And I don't know, in a classical ERT survey that we do, we have some like 1,000 measurements. Okay, so we do that, and then using Ohm's law, we just divide the current by the potential difference with some geometrical factor for the electrode arrangement, and we can get the apparent electrical conductivity. Using inversion, we can reconstruct the spatial distribution of the electrical conductivity, and this is the end result. A map, and it can be 1D, 2D, or 3D map of the subsurface electrical conductivity. Okay, so what can we do with that? Yes?
So the measurements are just on the surface of the
There are different types of measurement. In what I'm going to show here today, yes, all the measurements are surface measurement. But we can also do borehole measurement and to measure between boreholes. So there are many ways in which we can apply this method.
Yeah, but from the surface measurement, we get 2D or 3D picture of the soil.
Yes, can get like in typical setup. With the instrument that I have, a simple instrument, you can get to a maximum depth of up to 30 meters, something like that.
What is the scale? Special scale of what you see there?
So this is like a 24 meter, so tenths of meter, but this is really depend on the length of the line that you install. So what is the maximum distance between the electrode? Okay, so we get information or we measure and calculate the electrical conductivity. And using Archie law or the extension by Waxman and Smith, we can get important information regarding the subsurface properties and processes. So the electrical conductivity of the soil that we measure is directly related to the pore water electrical conductivity, or in other words, to the salinity. And people use this relationship, for example, to look at seawater intrusion to map to map the seawater intrusion. It is sensitive to porosity, to water saturation, to surface conductance if we have a lot of clay or metallic particles like pyrite. Those exponent here, the cementation exponent and saturation exponent, they are related to the geometry of the pore space. Okay, so if we measure the electrical resistivity of the subsurface, we can somehow know something about water content. And if we repeat, do time-lapse measurement, we can see the dynamic in water content in the rhizosphere. And this is the idea of using ERT, electrical resistivity, to look at water uptake by plants. And I'm going to show you here one example of how we use this method for root phenotyping. And this is Emmanuel, my PhD student. And the idea in this experiment was to use ERT to look at different root phenotype differing in their root architecture under water-limited condition. We did the experiment in the experimental farm in Rehovot under the rain-out shelter infrastructure that we have there. So no rain. We allow the plants to grow for a month, something like that, irrigating and with some rain. And after that, we did not allow any water to infiltrate into the soil. So we did electrical measurement. In this case, our line was 32 meter long with 0.5 meter electrode spacing. We took also supplementary measurement of water content, salinity, all kind of plant physiology parameters. We used ResiPy for inversion. I gave here some details about data processing. So after we take the ERT measurement, we do some temperature correction, some noise cleaning, and after the inversion, we get 2D map of the subsurface. In this case, you see the log of the resistivity. Using depth-specific calibration to water content, so with the TDR sensors, we measure the water content and we fit to the conductivity or resistivity at the same location, and got those depth-specific calibration to water content, which allow us to convert this resistivity map into a water content map. And we repeat those resistivity measurement for, I think that we have like 50 ERT measurements during the growth period. So almost daily measurement of the resistivity map. Now, an important assumption in this study, and the assumption is that change in water content is directly related to root water uptake. So we neglect Darcian fluxes, and the reason that we feel comfortable to do it is because we have no infiltration at all. So we started our measurement 10 days after the last rain event. So safe to assume that all changes in water content are related to root water uptake. Okay, and the result are spatiotemporal maps of root water uptake. And let's have a look at such a map. And this is what you can see here. So here you can see the depth. This is the soil surface. This is 1 meter below the soil surface. This is time in days. This is 60 days. And the colors represent the cumulative root water uptake from 0 to yellow, which is the maximum cumulative root water uptake. And you're going to see now 4 of those maps for the 4 different genotypes. This is for the WM203. This is the high biomass genotype with wide angle of the roots. So the roots spread horizontally quite a lot. And this genotype to reduce the highest total root water uptake, sum up to 750 millimeter. You can, of course, see that early in the season, the total uptake is low, and with time, of course, the cumulative root water uptake increase. You can see that most of the uptake is concentrated at the upper layer, and the more we go down, we have less uptake as expected. And this is the main thing that we can see here. When we compare this genotype to the other genotype, this genotype show really high intensity. So we take up a lot of water. The next genotype with high biomass and narrow root angle is WL140, and the total water uptake was just 500 millimeter for the whole season. So quite a big difference. You can also see that the strategy of uptake is different. So unlike the 203, here during those times it does not take a lot of water, but conserve water in the soil. So uptake gradually increase and also gradually shift to deeper layer. Okay? Now let's have a look at the low biomass genotype. So this is Galil. Here we have some measurement issues, so I cannot present the data. This genotype was with the lowest total uptake, only 350 millimeter for the whole season, compared to the 750, right? So it's a big difference. Most of the uptake, as you can see, confined to the relatively upper layer. And the last genotype, low biomass, wide angle, is the IPLR 760. And here we have moderate total root water uptake, 475. And I think that what is most interesting in this genotype is that we have relatively uniform root water uptake distribution. Right? You can see it here that it takes up water almost the same from all the layers. And just for comparison, you can see the whole 4 names. Okay, so let's have a look at the total root water uptake of the different genotype and the yield at the end of the season. And well, here you can see the WM203 with the highest root water uptake and also highest yield. But if we compare it to the 104 with only 500 millimeter of uptake, we can see that the difference, at least the average, difference is not that big. So if we think about drought resilience, then definitely this genotype can cope better with the water stress, right? And conserve water, keep more water in the soil. The Galil, despite the fact that this is the genotype that took the lowest amount of water, in terms of yield, you can see moderate yield. So it's not like the IPLR, right? The IPLR took the same amount of water as the 104, but the yield is really bad, really low. So we can see inefficient water use and really poor drought performance. Okay? So, I'm not a plant physiologist. This is not my objective. The main objective is really to show how we can use electrical resistivity to non-invasively quantify root water uptake at the field scale, and to show how using time-lapse ERT we can look at the specific characteristics of the different genotypes, of the different patterns of each genotypes in terms of timing, depth of root water uptake. What are the different strategies of those genotypes? And ultimately, the idea is to use resistivity as a tool for root phenotyping, because most of the breeding programs to make our crop better, more resilient, produce better yield and so on, they are based on the above ground part of the plant. And the below ground is almost not considered in those breeding programs. And we think that this tool can be useful to start thinking about root phenotyping at the field scale. Okay, so we saw that electrical resistivity is really sensitive to water content. It is also sensitive to salinity, but we cannot see the root. And if we want to see root, we need something else. If we want to look at the rhizosphere and what is going on in the rhizosphere in terms of chemistry, for example, or biological activity, we need something else. And of course, this something else is induced polarization, because induced polarization or spectral induced polarization, which is the method that I typically use, is really sensitive to interfacial properties and processes. So really sensitive to what is going on at an interface, for example, of a mineral and water, to the interface of bacteria cell and water, and to the interface of root cell and its surrounding. So before we dive into that, a few words about the induced polarization method. As before, this is an active method. We apply small oscillating current or field. This is the blue line here. And we measure the resulting potential, this dash line here. And as you can see, there is a phase shift between them. And this phase shift is related to polarization. Is related to the ability of the medium to store temporarily electric charge. Okay, so we again use Ohm's law, current divided by potential difference, some geometrical factor to go from conductance to conductivity, and we have the complex conductivity, because usually we represent those sine waves with in the complex plane using complex number. And this complex conductivity can be decomposed into the in-phase conductivity, the real part, and the qualitative conductivity, the imaginary part. And the in-phase conductivity is related to energy dissipation processes. So this is just heat dissipation, right? And really, basically the frequency range that we use in SIP, this is really similar to what we measure with the resistivity method. Okay, so this is really sensitive to water content, to salinity, and so on. On the other hand, the qualitative conductivity is related to energy dissipation processes, or in other words, to polarization. So what triggers polarization in porous media or around the root? And there are two main mechanisms at the frequency range relevant here, so up to 1 kilohertz. The first one is the electrical double layer polarization. So as you know, when we take a particle, right, most of the natural particles, they possess some net negative charge. And when we put it in electrolyte, a counter ion, cation in this case, attract, right, to compensate for this negative charge, and this is the electrical double layer. Okay, and this is under static condition. Now we apply some external electric field, and this external electric field cause ion to move and to polarize. So we have polarization, so we have accumulation of ions in one side, and if we remove this field, we have relaxation of this process, and this is what, in simple words, explain polarization in porous media at the low frequency range. And polarization of the electrical double layer depend on the ion mobility, so how strong those ions can move next to the surface, on the amount of charge that we have on the surface, on the size of the particle, right? If we have big particle here, we need to travel for longer distance, and polarization, it will take more time. Okay, so this is the main mechanism. There is another mechanism known as membrane polarization, and this is this polarization is due to the accumulation of charge due to if we have a semi-permeable membrane, for example, then we will have accumulation of charge near the membrane or in porous media. If we have pore throat, then ion cannot move, and we can have polarization. This depends, of course, on the membrane properties. If we have overlap between EDL of different particles, but it is important to remember that usually in porous media, the contribution of membrane polarization to the overall polarization is, well, most of studies talk about maximum 10% in comparison to the polarization due to the classical EDL polarization. Okay, so what we expect from root? Yeah, so root, the cells that compose the root, they have net negative charge on the membrane, and because they have net negative charge on the membrane, we can expect to see EDL polarization, like in every cell, actually, like we have in bacteria and so on. So let's see an example, and this is the work of my former PhD student, Kuzma Tsukano. And what we did in this experiment is to look at the sensitivity of SiP to the presence of root. We grow wheat again. This is a 2 months old wheat in hydroponic solution, and you can see it in our experimental column. And we did SiP measurement at different locations along the column. And we also measure the root biomass, the surface area, the diameter. So at the end of the experiment, we scan the root and measure and calculate those properties. Let's have a look at the result. So what you can see here is the in-phase conductivity. So this is related to the energy dissipation part of the complex conductivity. So this is the in-phase conductivity as a function of frequency for the different zones in the column. And, well, we don't see a lot of impact, right? It doesn't matter when we measure. What we get here is basically the logical conductivity of the water, of the electrolyte in which we grow the hydroponic solution. Okay, so no change. That means that we cannot expect to use ERT to monitor root. But let's take a look at the qualitative conductivity. So this is the qualitative conductivity as a function of frequency at the different zones. And you can immediately see that we have a monotonic decrease in polarization from zone 1, where we have most of the root, to zone 5, where we almost have no root at all. So monotonic decrease in polarization as a function of the amount of root that we have. And as I said, we also measure the root surface area, the root biomass, and so on. And this is what you can see in this figure here. You can see this is the chargeability. Chargeability is a measure to the magnitude of polarization. So this is the magnitude of polarization as a function of the total surface area of the root. And you can see a very nice linear relation between the magnitude of polarization and the total surface area of the root. Another feature of those electrical spectra is the relaxation time. This is the frequency at which we have peak in polarization at the low frequency range. And this is related to the polarization length scale, right? Like the particle radius that we discussed before. And we more or less see that we don't have big differences here in relaxation time, which means that the polarization length scale is more or less the same in every location. And actually, this is not really surprising for wheat, because in wheat, the structure of the root is like that, the diameter does not change a lot with age. So we expect to have similar root in different zones. Okay. So that was nice, and then we wanted to see, or to really prove, that the source of polarization is the membrane potential, the electrical potential of the cell membrane. So what we did is to kill the membrane. Okay, so we add cyanide to our columns. Then we have the columns with the hydroponic solution plant growing inside, and at time 0 we add cyanide solution and we monitor the SIP signature. And this is just a graph of the cell membrane potential as a function of time after the addition of cyanide. This is from a classical textbook, and you can see the drop in membrane potential. So we expect to see that the polarization vanish, right? And indeed, this is what we observed. Again, this is the polarization magnitude as a function of time after the addition of cyanide, and you can see this decrease in polarization, and that was really nice. And when we look at the relaxation time, then it started to be more complicated and interesting, because with time we saw an increase in the relaxation time, and that means that the characteristic claim responsible for polarization increased. And we thought, okay, maybe it is the root hair. Those are tiny, elongated structure, which is reasonable to assume that they will die first, and they are small in comparison to the main root. So this is an assumption, but we were not sure about it. And in general, we were starting to think about, okay, in the root we have the cells inside. So do we polarize the cells inside the root? Is it the root as a whole? What is the relevant length scale here? And we wanted to tackle this problem. Right, so again, is it the whole root that polarized, or the cells that compose it that we see? So to understand it, to try to understand it, we compose a numerical simulation and model in order to model the SIP signature of the root. So you can see here a cross section of root under the microscope. You can see the root cells. You can also see somehow maybe the root hair that I discussed before. And well, we took one of those cells and we made a simple model where this is the inner part of the cell. The cytoplasm is here inside. We have a membrane here, and we have here the extracellular space. Now to describe, right, so polarization, as I described before, electrical double layer polarization is the movement of charged species of ions, right, of those ions in the electrolyte or in the cytoplasm around a charged particle or body or cell. So we solved the Poisson-Lorentz equation, which considered diffusion and electromigration. We started with static condition, where we don't have any external electric field, so we just have the membrane potential, the electrolyte inside the cell, and the extracellular space. You can see here the potential as a function of distance. In this case, this is in nanometer, by the way. Okay, so this is the electrical double layer. You can see this is 1, 2 nanometer. Those are the scales when we talk about electrical double layer. This is the scale. So we have this solution of the electric potential as a function of distance from the cell under static condition. And then we apply our external electric field. So small field, so we consider, we use perturbation theory, so we linearized everything. And I'll not go into that, but eventually we got a solution of the concentration and potential distribution from which we can calculate the complex electrical conductivity. Let's have a look at the result. So I'm showing here the phase shift between the applied current and the measured potential, yeah, as a function of frequency for a few cases. This is the first case where we have a single cell in an electrolyte, okay, so one cell. Here we have 9 cells in the electrolyte, 16 and 25, and as you can see, polarization increase with the number of cells, okay? And this is as expected, as we expect. We have more EDL to polarize, so we have this increase in polarization with the number of cells. We also calculated here, this is the surface area of the root, of the whole root, right? It increased, the surface area increased, and you can see a very nice linear relationship between the phase shift at the peak frequency and the surface area of the root, like we have in the experimental data. So again, really nice to see some matching between this model and the experimental data, qualitative agreement. We did not try to model the whole root. When we look at the relaxation time, and this is the relaxation time, and I plot it as a function of the square of the diameter of the root, we got a very nice linear relationship obeying this function, which is what we expect from the literature, that the relaxation time is related to the square of the radius of the polarized body and divided by the diffusion coefficient. Okay, so again, very nice linear relationship, and this suggests that the main mechanism or the size that controls polarization is the whole root and not the tiny root cells. But in order to further examine it, we conducted another numerical experiment. So in this experiment, you can see here the way that we organize cell in the root cells in space. So we have 3 cells here. Now we have 6 cells, 9, and 12 cells. And each time we add the cells perpendicular to the field. Okay, so the electric field directly, or the cells directly exposed to the field. Here, 1 cell, 2 cells, 3 cells, and 4 cells. Okay, so we increase the number of cells directly exposed to the external field. And we did the same, but now we added the cells in a different way. So here we had 3 cells directly exposed to the external field. And we added 3 more cells, but the number of cells directly exposed to the external field is the same. And same here. And as you can see, no impact on polarization. So this simulation showed or proved that when we look at root polarization, we are sensitive to the root diameter, okay, and to the electric potential resulted from somehow the superposition of all cells inside. In addition, the relaxation time, something between 5 and 20 milliseconds, is reasonable when we think about the root diameter, millimeter, maybe a little bit less than millimeter. And if we would polarize the just the cells themselves would expect much lower relaxation time and also higher polarization, because then we would expect to have contribution from each one of those cells. And when people measure the whole plant, and they insert electrode into the root inside, they get those values. Okay, so we have some kind of screening effect of the root. Okay, so let's summarize. The key takeaways is that first, ERT enable us to non-invasively quantify root water uptake. And this really can change how we phenotype. Okay, so today, usually in breeding program, people think about the above ground part of the plant, and this gives a tool at the field scale to also look at what is going on below ground. Okay? SIP method extend the capabilities of ERT, allow us to detect root presence and physiological activity, and I think that future work is to link and integrate somehow ERT and SIP, so we'll be able to sense both physical processes and biological processes, again at large scales. And that's it. Thank you, and I'll take any questions. Questions?
So, if I understand correctly, the SIP method, you cannot use it with soil because you have polarization on the clay particles.
So
if I understand correctly, you cannot use polarization when you have soil particles because you have polarization also on the clay particles.
So to differentiate between the signature from the root and the soil.
Right.
Yes, this is a correct observation. I'm not sure that we'll be able to use SIP to look at root in soil, but only in hydroponic. And we did a few experiments and published a little bit about it. And the thing is that maybe we cannot see the root, but we can see the rhizosphere. Because the rhizosphere, the soil around the root, is a hot spot with really distinguished chemical, biological, and physical properties. For example, root exude mucilage, which is a polymeric gel, and this really affects the biological properties of the soil. And we can see that, but not directly. Yeah, not here, not in this model. Of course, we can consider evaporation or even Darcian fluxes, and not to go with this direct coarse approach, and go for example, in this modeling of Richard equation with sink term inside. Yeah. This is what we assume. Indeed, we assume no evaporation, no percolation, and this is a reasonable assumption under the condition of the experiment, because in this experiment, we started it 10 days after the last irrigation. The upper layer of the soil was already quite dry, so evaporation is not that big. We are at, I don't like this term, field capacity, but we don't have, if at all, percolation or redistribution of water in the soil. This is, and in comparison to the large amount of water taken up by the plant, this is, I think, reasonable assumption under these conditions, of course. Yeah.
You have a plot of the polarization magnitude as a function of the surface area, and you said it's into a linear, like a linear fit. You said it looks linear, but it actually doesn't look entirely linear. It looks like an increase in maybe a shoulder. Would perhaps a different function describe it even better?
Perhaps.
I mean, with the relaxation process, you can fit it with exponential decay factored in 1 minus an exponential decay.
Yes, but I would be quite careful with trying to give. Those are complicated experiments to do, and even to extract this chargeability. So to do that, we fit those SIP curve, the spectral response to cold model, for example, which is highly non-linear. We try to be very conservative in that perspective, and not to over-interrupt this with some, unless we know the physics.
Can you use the conductivity method in a microscale, like look what happens inside soil aggregates?
So first, yes. And a colleague of mine, they try to build an instrument in which you can measure the conductivity of one particle. But I would like to say something else actually. And this is, for me, this is one of the most exciting things in this SIP method. And this is the sensitivity to the nanoscale processes. So we are sensitive to what happened in 5 nanometer, 2 nanometer electrical double layer. And I have, for example, another work in which we look at the impact of flow on the SIP signature. And using SIP, we're able to start thinking about flow at the electrical double layer, about chemical complexation, absorption and desorption processes at the electrical double layer. So this method is really sensitive to the interface and what is going on in the interface. And we do the measurement at a large scale, but really sensitive to the small scale. So this is what I really like in this method.
I have a question. Can you go back to the first part where you were showing the different the uptake at the root level? Yeah. So just a kind of probably stupid question, but do those maps also correspond to the biomass of the root in the surface, or was there just different functioning of the roots?
So those maps are just the cumulative root water uptake. This is what I showed here. We tried, and we were not successful in trying to measure root biomass below each of those plots. And this is a tedious and really, I mean, anyone that report root length density from the field, I would really, I mean, it's so difficult to separate between the tiny millimeter scale, sub-millimeter scale sometimes, roots and soil particles, and especially, I have a different work in which we look at root at the pot scale in the greenhouse, at which in this work we look at root water uptake. But again, this is in the greenhouse with really confined geometry, so we're able to look at the root length density and to measure the root. And actually, the results were quite interesting because we saw that under water-limited condition, we don't have a good agreement between root length density and uptake because we have hydraulic compensation. If some area is dry, and for example here, this area is going to be dry because most of the water are taken up from here. But we have most of the root going to be here. Still, most of the water late in the season will be extracted from a different location. And actually this is important because in many ecological modeling, root length density is an important parameter that responsible for transpiration.
Any more questions? So stand up speaker again.